Hello everyone. Now we are going to discuss about chemotherapy and antibiotics. Before entering into vast area, we should know some technical terms, that is chemotherapy. What is chemotherapy? Here, by name itself, we can easily understand with the help of chemicals we are going to treat some diseases. So what is chemotherapy means? The treatment of diseases by means of chemicals that have a specific toxic effect upon the disease producing microorganisms or which is selectively destroy cancerous tissue. Then the another important term is antimicrobial agents, compounds that are used to kill or inhibit the growth of microorganisms. Next important term is antibiotics, the substances produced by some microorganisms that can kill or inhibit the growth of other organisms. Then antibacterial refers to the substances that can act against bacteria. Paul Ehrlich, he is the important scientist in the field of chemotherapy because he discovered salvarsan 606 is a kind of a derivative compound of arsenic. Sometimes it is otherwise called as magic bullet. This salvarsan 606 is capable of destroying spirochetes of syphilis. So he is called as father of chemotherapy, and his invention leads to rise a new branch of medicine. Next important discovery in the field of medicinal science is antibiotic. Alexander Fleming is the important scientist in the field of microbiology. He accidentally discovered penicillin produced by fungus penicillium. How he discovered this penicillin is interesting story. While working in lab with staphylococcus culture on an agar plate, meanwhile he went on vacation and he came back after 2 weeks and he found the growth of some mold in staphylococcus agar plates, and he noticed the mold growth prevent the staphylococcus bacterial growth. So this is an incident that leads to the invention of first antibiotic. This is some important terminologies related to antibiotics. First one is antibacterial spectrum. This will denote the range of activity of an antibiotic. Second one is broad spectrum of antibiotic. Broad spectrum means some antibiotics have wide range of activity. Means they can inhibit wide range of gram-positive as well as gram-negative bacteria. Next one is narrow spectrum antibiotic. Narrow spectrum means it will active only against limited number of bacteria. Another important term related to antibiotic is MIC. MIC stands for minimal inhibitory concentration. The lowest concentration of antimicrobial that inhibits the growth of bacterial population. Here we can differentiate bacteriostatic and bactericidal antibiotic. Bacteriostatic means the compounds which will inhibit the growth of microbes, where in case bactericidal means the compounds which kill the microbes. Example for bacteriostatic activity is chloramphenicol, erythromycin, clindamycin, tetracycline, etc. And for bactericidal activity, beta-lactams, vancomycin, rifampicin, metronidazole, etc. Here we are going to discuss about mode of action of antibiotics. Mainly antibiotics have 5 different mode of actions. They are interfering with cell wall synthesis, acting on cytoplasmic membrane, inhibiting protein synthesis, inhibiting nucleic acid function, metabolic antagonist. First kind of mode of action of antibiotic is interfering with cell wall synthesis. As we all know, bacterial cell wall is made up of peptidoglycan, uramic acid and other molecules. Cell wall nature and the structure is mainly depends on peptidoglycan layer. In gram-positive bacteria, peptidoglycan is tightly packed, so the gram-positive bacteria cell wall is thick layer in nature. Whereas in gram-negative bacteria, peptidoglycan is loosely packed and its cell wall nature is thin. Beta-lactam bactericidal drugs, they inhibit bacterial cell wall peptidoglycan synthesis in growing bacteria. This leads to death of bacteria. Next cell wall synthesis interfering antibiotic is vancomycin. They kill bacteria by interfering with peptidoglycan polymerization. These are all some antibiotics which will interfere with cell wall synthesis. They are penicillin, cephalosporin, bacteriacin, vancomycin, and cycloserine. Second mode of action of antibiotic is acting on cytoplasmic membrane. Certain antibiotics bind to cell membrane of that bacteria. Semi-permeability of membrane is lost because of this attachment. So example for those antibiotic which will act only on cell membrane is polymyxin, nystatin and amphotericin B. Third kind of mode of action of antibiotic is inhibition of protein synthesis. It will mainly inhibit translation process. Some antibiotics act on 30S or 50S subunits of ribosome. Here it will affect initiation, elongation and termination of peptide chain formation. Here for 30S some examples are there, and for 50S erythromycin, chloramphenicol and lincomycin it will only affect the 50S subunits of protein synthesis. Fourth type of mode of action of antibiotic is nucleic acid synthesis inhibition. Here the inhibitor of RNA synthesis is rifampicin. They kill bacteria by inhibiting RNA polymerase. For DNA synthesis example is novomycin. It will inhibit DNA replication. Metronidazole damage DNA and inhibits replication. Quinolones block DNA gyrase enzyme activity. Fifth one is metabolic antagonist. Their structure resemble to essential metabolites which is usually produced by bacteria, thus can compete with them. Example for that metabolic antagonist is PABA, para-aminobenzoic acid. It is a main cofactor for bacterial cell growth. Sulfonamides, structure which is exactly similar to para-aminobenzoic acid. Other examples are sulfones, trimethoprim, etc. Thank you.